Hello there, it's Mrs. Scoville, and I'm back today. We're going to be talking about independent assortment of multiple alleles. We are dealing with genetics, so this is going to be a little more complicated today. There are multiple objectives for today's lesson. We have three. The first is to state Mendel's laws of inheritance. The second is to find all possible gamete combinations when given a genotype with two alleles. Yesterday we only had one. And to complete a Punnett square for a genotype with two alleles and find the phenotypic ratios. So this is going to be quite a lot. Okay, so what we're going to start with today is a little bit of a review, and also I want to emphasize something I didn't emphasize in yesterday's lesson. So we're going to start out just reminding you that we did monohybrid crosses yesterday, which means we were just dealing with one trait. And in this case, the example we have, the trait is seed shape. And you might be wondering, why are we dealing with seed shape so much? Well, this goes back to Gregor Mendel and all his work with pea plants. So those are used very commonly. So the alleles that we have here are big R for round, so because it's capital, that's our dominant one, or little r for wrinkled. So that's the recessive one. If we cross two heterozygous round seeds at this point, I'm confident that you guys are good with Punnett squares. So, you just break up the alleles for the parent and for the two parents. You put one on one side, one on the other, one in each box, and then you just match them up. So in doing this, we know that we can do probabilities and ratios, but the one ratio I didn't have you guys do was genotypic ratios, so we're going to look at that very briefly. Okay, so as a reminder, what we did look at yesterday very often was the phenotypic ratios. So if we look at the phenotypic ratios, our two options are basically round, which is our dominant, or wrinkled. So you'll do round to wrinkled, actually make those dots, round to wrinkled in your ratio. So if we look, three of these are going to be round because they contain at least one capital R, so R anything will be round, whereas wrinkled will only be little r. So here, if we look, it's going to be 3, and I don't know why I'm putting equal signs, but it should be dots, 3 to 1. Okay, so that would be our phenotypic ratio. When you look at them, you'll see 3 that are round and 1 that are wrinkled. The genotypic ratio, on the other hand, so genotypic, that looks terrible, but that's okay. Genotypic ratio, there you go from all dominant, so you go from homozygous dominant, so homodominant, to heterozygous, to homozygous recessive. So if we look at this one, we've got 1 that's homozygous dominant, 2 that are heterozygous, and 1 that's homozygous recessive. When you cross two heterozygotes, that's always going to be the genotypic ratio, 1 to 2 to 1, and the phenotypic ratio will always be 3 to 1. So I did want to just remind you guys of not only the phenotypic ratio, but show you how you do the genotypic ratio as well. Again, you go from all dominant to all recessive, and the heterozygotes will be in between. Okay, the other thing that I do kind of want to emphasize here is just really make Mendel's laws clear. You saw it yesterday in the little brain pop video, but here, basically what Mendel found is that when gametes form via meiosis, and remember that's going to be egg or sperm that forms, those alleles that we have, for example, the big R and the little r, are going to be separated from each other. And that's just going to happen due to independent assortment. So later, when fertilization happens and egg and sperm come together, this is going to give you the genotype that you have in the offspring. And that's also why we break it up when we do the Punnett square, because they're going to get either big R or little r in the previous example. So again, if we just look at this one more time to show you what I'm saying here, if mama here has big T, little t, for tall and short, this is plants again, the big T and the little t can't be in the same female gamete, so those are not going to be in the same egg. They have to separate because in meiosis we know it goes to half, so it's only going to get 1 copy of each. Same thing with dad, those have to be separated in the sperm. And if you're starting to question, like some people have asked me before, well, how do plants do this? Plants aren't doing sexual reproduction. Yes, they are. It just looks different. So pollen in the air is basically plant sperm. Okay, so then getting back into the law of independent assortment, so the law of segregation happens because of this, but again, what this explicitly says is that those different traits, those different alleles are going to be separated from each other in the egg and sperm, as we saw. But we can actually illustrate this using something called a dihybrid cross, which is going to be the focus for the most of the rest of our lesson. Okay, so dihybrid cross, that sounds very similar to monohybrid cross, except di means 2. So here we're going to look at the possible inheritance probability of 2 traits. And in Mendel's law of independent assortment, remember, again, one more time here, that the individual alleles are going to separate during gamete formation, which is meiosis, okay? So during meiosis or meiosis, those alleles are going to separate from each other. The thing that's interesting is we can actually predict how many possible combinations you will have based on how many alleles you're looking at. So here we've got two different possible alleles, and that's going to be 4 gametes because we're dealing with 2 alleles. So 2, you do 2 to the n, so 2 to the second power, that's 4. Here we have possible of 3 different gametes, or sorry, 3 different alleles, because look, see, there are 2 of them. So 2 to the third, that means there are going to be 8 different gametes. So this isn't something that's super important for you guys to do, but I did want to let you know you can actually predict based on the alleles that are present how many possible different gametes you will have. What I do want to point out to you though here, some of you are still struggling with the idea that meiosis, not only are the gametes not similar to the parent cell, but they're not similar to each other. So again, I did want to point this out because all of these gametes are different from each other. So very few of them are going to be identical. Okay, so now we're going to go through doing the actual dihybrid cross, and I kind of broke it down into 3 steps for you. This is going to take a little bit more work, just because, first of all, we're dealing with 2 traits. So in this case, we're going to look at seed shape and seed color. We saw the seed shape earlier, round or wrinkled, so that's going to be capital R for round or dominant trait. And then we're also going to have yellow and green. So we can tell here, because it's capitalized, that yellow is our dominant trait. We have 2 heterozygous round yellow plants that we're going to cross, and we want to know the possible gamete combinations. So how you're going to do this is first you need to just go ahead and actually write out the genotypes based on what they've told us. So it says they're heterozygous round and yellow. So, and they're both that way. So once we figure out one, we'll figure out the other. Because it's round, I know it's capital R, and it says it's heterozygous, so that means I need to put a little r. Same thing, it just said it's heterozygous round and yellow. So the yellow is going to be big Y, little y. It says they're both like that. So I'm going to do big R, little r times big R, little r, big Y, little y. So when people write these, you just literally put it as like times because it's a cross. So how we'll figure out the gamete combinations now, we're going to employ basically the FOIL method, very similar to what you learned in math. So it's first, outer, inner, last. We're going to do that very similar. So our possible gamete combinations, because when the gamete forms, it can either have big R or little r, we need to match up the r's with the y's. So our first possible gamete combination is going to be big R, big Y. Again, you can only get either big R or little r. You can't have both. Then big R with little y. That's another possibility. So that's one. That's a second. Now we need to match up the little ones. So little r with big Y and little r with little y. That's our last one. So you basically just match them up. And again, you can only get either the big R or the little r, either the big Y or the little y. So you're just matching up to figure out the possible gene combinations. And this one, because it's the same, it's going to be exactly the same. But again, if we just match them up, big R, big Y. Second one, big R, little y. Little r, big Y. And then finally, little r, little y. So basically, this is showing us all the ways that the gametes could be mixed up. Next, again, that's our first step, just figure out the gamete combinations. Once we've done that, we can go ahead, and this is going to be a 16-box Punnett square. So instead of just 4, we've got 2 traits, so it basically quadruples the Punnett square. So you'll write those in, and these were all our combinations we came up with on the previous slide, and now I'm going to show you how you'll fill it in. This is very similar to what we already did. You always write the first trait, though. So big R was first, big R. Y is second, so you still write it second. So you put both of the R's in before you put the Y's in. So if we do that here, big R, big Y, little y. And remember, still you follow the rule of you put the dominant one first. It doesn't matter which parent it comes from. You just need to fill it in dominant first. And so you see I did that again. I've got a little one, big R, little y, little y. So you just go through and match them up. Big R, little r, and these are 2 little y's. So you're just matching them up just like we did before. Big R, little y, big Y, big Y. Big R, big Y, little y. So you do have to pay attention as you're doing this because you're going to see a lot more different gene combinations on this one than we saw in the previous one when we just had a monohybrid cross. So now these are all filled in. So now we've written in our allele combinations. That's the next step. So we found our gene combinations. We got those listed on the top. And notice here I had 4 gene combinations, so you just write one by each box. Okay, now we can figure out the phenotypic fractions and ratios. So this is just a different view of it. These are all the genotypes that we came up with, but I like this because it helps you visualize. So here, any of them that have at least one big R, we know that those are going to be round. So big R anything, they're going to be round, and big Y anything, they're going to be yellow. So when we do our phenotypic ratios, we're going to have some more options here, because again, we go from all dominant. So the all dominant would be round and yellow. And then you go with the first dominant, so then round and green. And then you move to the yellow again. So then we're going to have wrinkled and yellow. And then wrinkled and green. So what I'm doing here is I'm going from all dominant to all recessive again. And so these are all recessive. And in the in-between, you just go with the first dominant, second recessive, and then here you go the first recessive, second dominant. And what I mean by first and second is just the trait. So the round was listed first, so I need to put that one first. So now if I count how many are going to be round and yellow, well again, if it has big or anything, it's round. Here I visualized it for you so we can easily see, but it's going to be 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 will be round and yellow. The ones that will be round and green, it's going to be these 3. And again, they're round because they have big R anything. Green is going to be the little y, like we have right here in all 3 of these. So it'll be 9 to 3. The wrinkled and yellow, so to be wrinkled, they have to be little r. So those right there. And to be yellow, they just have to have a big Y. So we've got 3 of those as well. And then we're only going to have one, the little r, little y, that's all recessive. So 9 to 3 to 3 to 1. Anytime you do a heterozygote to a heterozygote, like we just did, it's always going to be 9 to 3 to 3 to 1. If I asked you for any of these in fractional form, well, you already have them. It's 9. There's 16 total, so that would be 9 out of 16. Same with all of these, 3 out of 16, 3 out of 16, and 1 out of 16. So if you're asked for a fraction, well, you just count how many of, the box. Okay, so we're going to go through one more example very quickly. We've got a homozygous round green plant. Because it's homozygous round, I already know that it's going to be big R, and little y. I'm just going to write that up here. So that's big R, little y. And I knew it was homozygous round because it told me. So that's the big R. The little y, that's the green. So these, my possible gamete combinations, well, there's really only going to be one, because if I do first and outer, those are exactly the same, and these are going to be exactly the same as well. So when you get into the homozygous, then you have a lot that are the same. So these are all going to be big R, little y. So I'll write those in. The other one, it's homozygous recessive, so those are going to be all lowercase, and so little r, little y. Again, you have to match up one R with one Y, so these are actually all going to be the same as well, but they're little r, little y. Because if you notice here, these all look exactly the same, we're only going to have one genotype here, and it's going to be the same through all, so I'm just doing that to save time. If I was actually asking you to fill this in, I would want you to write all of them. If we do our phenotypic ratios on this, though, you still need to go from all dominant to all recessive. So this time, just to help you, I'm going to write out what the phenotypes would look like. So I'd be looking for round and yellow for the all dominant, and then round and green. So the first dominant, the second one recessive. And then I would go wrinkled and yellow. So the second one is now the dominant one. And then finally wrinkled and green. So if I count these, well, how many do I have that are round and yellow? These, that's going to be round, but that is going to be green. So I'm going to have 0 that are round and yellow, and all of them, all 16, are going to be round and green. None are going to be wrinkled and yellow, and none are going to be wrinkled and green. So in order to have wrinkled and green, I would have had to have little r, little y. In order to have wrinkled and yellow, I'd have to have little r, big Y anything. Here, my round and green, big R anything, little y, which is what I had. And then here, big R anything, big Y anything. I'm saying the anything because that could be another big R, or it could be something else. So same thing here, basically 16 out of 16, 0 out of 16, that's how you would do the fractions. That's it for the lesson. Read your summary. Have a great day.